So this is going to be very much workshop-y, interactive. We have a couple of mics in the room. Try to use the mics. If you have questions or you want the demonstrator to try something different or whatever. And I don't really have a vision of how well or poorly this is going to work. The software could crash. You know, so these are not pre-prepared. Well, they're a little bit pre-prepared, but these are not slide decks. So this is real-time demos of real software written by real people who are sensitive to their code working, so try to be nice. And this is Khaled Alganem. Khaled started as a Master's of Mathematics student last year, and at the end of his first year, his advisor said, hey, I'm going on sabbatical. This would be a great guy to have in the PhD program. So I broke like 50 University of Toledo rules and stole him for the neurosciences graduate program. So now he's a first-year neuroscience graduate student. Phenomenal coder, I'm told, because I don't code. And the application he's going to present, his name is Khaled. I named it Kaleidoscope. Initially, it had an H in it. We took that out because that was too personal. And the genesis of this is in December of 2017, when I was at the University of Cincinnati, I had a graduate student finishing. Both laser capture microscopes broke. Her last chapter of her thesis was to cut cells out of an animal model of schizophrenia and do confirmatory studies. And so we were really dejected, and I said, you know what, let's do bioinformatics. And we started doing manual data mining of schizophrenia databases and tried some things, and she did a beautiful paper on that, and it's all published, and there's a drug study, we're not going to get into that now. And then the idea was, well, could this be automated? And that's, I'm just going to hand it over to you, and that's Kaleidoscope. So just show us what you've got, show us what you've done, but you have to tell us what it's for.
Sure. So yeah, thank you. So yeah, this was my first project when I joined the lab last August.
You have to use the mic. You have to use the mic.
Yeah. So at the beginning, it was just to automate the lookup publication studies from schizophrenia databases. But over time, that software evolved, and now I think it's called bioinformatics platform, where you can have access to computational tools and genomics and just transcriptomics data sets, where you can look up anything you are interested in, specifically target genes. I like to call it the exploratory data analysis platform. So you can utilize great and powerful tools and huge biological databases. So yeah, the idea was we know there's a lot of databases and that's considered underutilized. And the idea of this software is to have these databases in one place with an easy user interface where you can look up, you have some questions and you have the interactivity of the software so you can get results very quickly. So I'll go over the databases we have so far quickly. So the first one is the brain RNA-seq. It's RNA-seq of like single-cell RNA-seq, isolated from human and mouse cortex tissues, and this is cell-type specific. Also, we have the brain cloud database. It's also from the cortex. It's transcriptome across the lifespan of the human, so you can see the genetic control transcriptome of the targets over the life. Also, we have the GTX, genotype tissue expression, where you can see the expression levels in different tissues in the body. We have the brain atlas. It's a huge database for the brain. It gives you also single cell RNA-seq data, but it's more involved in like we have more clusters of cells. We have the inhibitory neurons and side neurons that as well. Also, we have string. It gives you protein-protein interactions. So if you start with just one single target gene, it can expand that target gene. Gives you protein that interact with that. And then we have the lookup, which is the foundation of the software, which you can look up previous studies and diseases, specifically we are working in schizophrenia. So we looked up schizophrenia databases. And then we have also connected to others as well. And then also we have the GWAS catalog, which is one of the biggest GWAS databases. It gives you genetic variants and phenotype association as well. So I was planning to start with a target gene myself, but I thought it's going to be interesting if somebody give me a target gene to start demoing the software. So if anybody has a target gene that we can use to demo the software, that would be great.
We need a volunteer. If you know the Uniprot team, that helps. I didn't plant anyone in the audience. Does anybody have a favorite?
GT1, 2B, or
What would that? I don't want that to be SLC A12.
right? Something, yeah.
So we
check.
Or do we want to do a cancer gene?
Maybe cancer. Okay, whatever.
Come on, man. No, he's right.
Yeah.
What do you want?
Yeah, do that. So but I don't know the full Uniprot name.
yeah.
Isaac's looked it up for us. Yeah, I'm totally biased to GLT1. The GLT1 is an astrocytic glutamate transporter.
182.
An A2.
SLC 1A2. So it's an A.
A2.
A is an atom.
Yeah, so this is the brain RNA-seq. It's going to give you the expression levels on different cell types of the brain for mouse and human. So yeah, this is, so we have the mouse here, and then with the human, and this is the FTK values, which is like a fragments per kilometers per million. It's just kind of a value for transcriptome expression levels.
Yeah, so when Yusuf comes to my office and says, hey, I want to study this in endothelial cells, I say, okay, let's check. Maybe we shouldn't do that, right? Yeah. So this is sort of a validation check we do when anybody comes to us and says, hey, I'm interested in this treatment, especially in the brain. Keep going.
So the chloride channels?
It was just the gene.
It's very nice.
Same address.
Then we can go to string and then expand that just one target gene, and then we can get the interactor the proteins for that target. So this is based on known interactions or predicted functional association with the target. So yeah, the edges is based on either known interactions.
Can I ask a question? What website are you on right now?
What's what?
What website are you on right
This
now?
is
How
the
do I access
Yeah,
this?
this is the string. Oh, this is my
This
you
is
mean
your
the whole
platform.
thing? Yeah. Yeah.
Okay, so we'd have to actually go into string or go to NAVs to do the analysis ourselves.
No, you can
So-
go to the
Go
web
ahead.
page, this web page, and you can have access to all.
Is this free?
Maybe
Yeah,
I,
this is
maybe I didn't
No,
read
so
the
this
website.
is part of the Khaled's genius. This is a pipeline app. So you can go to the String website and run String, but Khaled's software does that for you. And it does that for you for all of those online databases. Your arc is a comment.
But is it common? This is Shiny app, right?
Yes, this
Our
is
Shiny. And it's similar to what you will see for SQL. So this is a platform that can be easily deployed as long as you install R and R Studio, which are open source programs on your computer, any laptop or server, and you can run it. It's essentially providing browser level access to computational layer, server layer, which is R, and connectivity to other sources via API. So that is completely portable, and so as this is available somewhere on GitHub, anyone can download it around.
yes.
And aren't we hosting it?
Yes, it's hosted in a server. You don't have to download anything for that. Just go to the web.
It's not working on my computer for yours.
I got it up on mine. The URL, I think, is and you have to put every single thing in perfect, including like backslashes for it to work for me.
And case sensitive too?
I didn't work for me until I capitalized for 8 databases, and then it worked, but I couldn't
Okay.
finish the concept.
I can send you the link after the talk.
We are happy to share the link. That coders live for people using their code. At least that's what they taught.
Yes, can you go back to where we were just like now,
The strength.
to the seed sequence where you have the networking
The string, anyone see the
string, yeah.
So what he's done here is you can set the score, which is the strength of the interaction, and the number of nodes is the number of proteins you throw up. And the red dot in the middle is GLT1, right? Yes.
And they,
So can you click there? What do you do with those networking? Can you?
you can, see the color of, the color of the edges can tell you kind of interactions, if it's non-interaction or predictive interactions. Also, you can go down and see the list of the targets, and then you can have a description for each target as well.
Nice.
So yeah, keep going.
That's great.
I got it to work. I was putting LO instead of IO.
So we have validation.
Yeah.
So while he's loading this next part, so this is a pipeline app. So this is brain biased, but this brings together. Go ahead and run it. All of the tools that we've been using in our laboratory. I'll shut up and let you explain this.
So this is based on the GTX database. It gives you expression levels and different tissues in the body
So
for the whole network.
you copy.
Yeah, exactly. The copy of the network from the string, and then put it here, and then you can see the expression levels in each tissue.
Very nice. Yeah.
So
We have
again,
these different tissues.
Yusuf says, I want to study this.
That's
I want to study GLT1 in epithelial cells. It's not expressed there, but 23 of its nearest interacting proteins might be.
Save a lot of time.
Well, before I buy an antibody for a summer student, I'm looking at this.
Wait.
Before we even touch a pipette, we do this for every project.
Yeah.
Keep going, Khaled.
So we take that as well to the brain atlas. This is single cell RNA in the middle to lower gyrus. This also gives you expression levels in different cells. You go to the heat map. You can also see the expression levels in these cell types. So we have astrocytes in the endothelial, cytid neurons and immune neurons. So you can see where the target genes are expressed more or rich in specific cell level. Now we have the lookup page. This is where the software started with the lookup replication studies and previously published data sets. So we have these models of diseases we have. So since our lab focuses on schizophrenia, so we have extracted previously published data sets that related to schizophrenia, and then we can compare the expression levels of these targets and control against the schizophrenic patients.
So what we realized is that someone does RNA-seq on their specific experiment, and they care about 12 genes, and to publish it, they have to reposit it somewhere. So we swoop in, access that database, and I think we have 14 or 15 full change schizophrenia versus control experiments loaded. And while he's pulling that up, Khaled has written this so that it's scalable. Anybody can add any full change database that's in the right format to this. And Khaled's worked with other people in our laboratory to build a dopamine database of if you perturb dopamine in some way, a knockout and knock in, any kind of RNA-seq data, they're building that. We're building an MDD database.
This is also customizable, so if you interested in specific disease, you can upload your own RNA-seq data sets and you can compare. So you're not limited to these data sets. So when you get the results, you can see the fold chain values for each data set for control against the schizophrenia.
It shows a heat map.
Oh,
that's cool.
Is this a kaleidoscope?
This is Kaleidoscope,
Is this a kaleidoscope?
named after Khaled. They get credit for it. Or blame.
So yeah, this is the list of genes that we have from the strain network. And the bars represent the there's so many people accessing the program at the same time.
Yeah.
That's why.
Yeah, we're hosting it, but not very well apparently.
So it gives you
Yeah. I try for your heat map right away. There you go.
Yeah, so this is a heat map for the low fold change between control and schizophrenia for each target gene that we got from the network, and for each data sets that we put for the NVIS schizophrenia module. So a red means the fold change with the positive values, which means that it's upregulated in schizophrenia patients compared to the control.
So even if Yusuf's favorite hit is not regulated, or maybe it's not that interesting, what about 24 or 5 of that protein's nearest friends based on PPI interactions, right? And you could also game it towards certain pathways and so on and so forth. And basically, we're trying to improve our guessing before we go down the rabbit hole following a specific hypothesis, because studies of postmortem brain are expensive because the tissue is hard to get, and all the other challenges. And that's really where this came from. Is there anything else you want to show us on this?
I mean, here in the purple.
Or
General.
yeah, go keep going.
So we go to Alex. Since we have this network with 25 targets, you can see how many knockdown genes are available in Alex.
So I want to say something about that. So there's lots of ways. Dr. Meller's the very next person up and is going to demo iLinks. There's lots of ways to interface with iLinks. One of them is you have an RNA-seq signature. You can extract your own L1000 and just have at it directly. Another way is you don't have RNA-seq, but you have target genes, and you can look in iLinks, like Dr. Meller mentioned earlier, for a knockdown signature. So GLT1 or whatever SLCA1A2, I don't think has a knockdown signature in iLinks. So that was depressing for us. But glutaminase does, which is GluL. So there are things we can do, and there's other ways to get around that too, and Dr. Mellon mentioned those as well.
So yeah, this will give you more details for the gene knockdowns. So we have the target genes and the cell line side, and then we have a link, can go access Alex as well.
Yeah, it goes directly into iLinks, pulls up the page that you're interested in for that knockdown, for those concordance scores, for those drugs, those perturbations.
So we also have this GWAS catalog database. Like I said, this gives you genetic variants and phenotype association. This is a huge database with about over 4,000 studies or about 200,000 SNPs and phenotype association. So it gives you what kind of SNPs. So we have these target genes and the SNPs and the context of SNP and the p-value, and also a link to the actual study where you can actually go and read about the study. And this gives you the most common phenotype association with the network.
And that's sort of a validation, right?
Yeah, see the first one is schizophrenia. Also want to see if what kind of genes and that network kind of drive that phenotype. Pretty short up. So we have this list. So we have the top common phenotypes, and then we have the list of the targets, or what kind of sniffs are.
So Khaled and I have done probably 50 Skype calls in the last year with collaborators demoing this software. Some with Scott Maruzzi, he's over there too. And it's pretty easy to use, I think. We're hosting it. If it gets too popular, we'll get in trouble because I don't know how well we're hosting it. I don't know how much hosting capacity we have, but we're hosting it. And it's been really fun developing this. And we've gone from manually curating across 14 databases, looking at one gene at a time to see if it's changed, to just hitting a button and having access to all these resources, including iLinks and String and the GWAS database, all in one go. It's really an awesome job, Khaled. Is there anything else you want to show us?
Can I ask a question?
Yeah, absolutely.
Do you recommend when we're trying to learn how to use this to do one at a time, or to actually do a whole family like you're doing?
I mean, if you know how to use just one, you're gonna do the same thing for the others. So
I mean, I guess when it comes up to talk about iLinks, my problem with iLinks is the data's overwhelming. It's hard for me to know what to, you know what I mean? There's so much there. It's hard for me to know where to go with it. Like, it gets tricky if I have, maybe that's just me. No, that can't just be me. People get overwhelmed by data, right?
Yeah, so I'm going to defer to Yarek, who's up next. In my laboratory, we've had to have iLink super users. So the graduate student who worked on this, Courtney Sullivan, was an iLink super user, and she spent a lot of time with Yarek and Jim Regal and others in Dr. Meller's group. And so, however, for Kaleidoscope, we don't view this as needing a super user. This is something we're planning on teaching to our rotating undergraduate students this summer, with supervision and help, of course. And it's really been designed to really, as a pipeline act, really bring everything together, be super user friendly. And so we always call it up if it glitches at lab meeting, whatever. Justin, you have a question? No. Any other questions or comments for Khaled? Let's clap for him.
For our next session.